Hi everyone, thanks for coming to my talk. I'm Kelly, and today I'm going to be presenting joint work by the Kandaji lab at Stanford and the Mahoney lab at Penn State, predicting transcription factor binding across species with neural networks. So to begin, why do we want to do this? Well, simply, if we had a series of chipstick experiments in one species and we could impute them into lots of other related species, that would save us a ton of hours and time that we'd otherwise spend redoing those experiments in every species of interest. Many TFs and their high-level gene regulatory programs are highly conserved across mammals, and in particular, if we look at the PWMs representing the sequence affinities of those TFs, they are pretty highly conserved, and so we do expect the logic of TF binding to generally transfer across species. Interestingly, though, if we look at parallel chipstick experiments across species and look at their individual binding sites, there is remarkable divergence at the individual binding site level, indicating that we're not going to be able to use a very simple alignment approach from one genome to another in order to do something like this. Instead, we're going to need more sophisticated models. Importantly, if we did have models that were accurate and could do this well, we could learn some really interesting novel insights potentially into the evolution of gene regulation across these different species. Our initial strategy was that we binned the mouse and human genomes into 500 base pair windows, and we labeled every window with a 1 if a ChIP-seq peak fell within it and a 0 otherwise. We trained one model on mouse liver ChIP-seq peaks and one model on human liver ChIP-seq peaks, and then we assessed the performance of those models on held-out data from both the training species and the cross species. We did this for 4 TFs, and we used a model architecture that's similar to what was state-of-the-art for the ENCODE DREAM challenge on cross-cell type transcription factor binding prediction. So briefly, the input to this model is a 500 base pair sequence. It's a hybrid convolutional recurrent model, and the output of it is a 0 to 1 probability of whether or not a TF is bound at that site. So jumping right into results, when we assess how well these models do on both of our test sets using the area under the PRC, what we see is that the model that is being tested in the same species that it was trained on always outperforms the model that is doing cross-species prediction. This isn't much of a surprise, but in some cases, the gap between the two models is pretty huge. And so what we'd like to do now is investigate what's going on and see if there's any way that we can help lessen this gap. So what we're going to do is try to figure out which loci are being differentially predicted between the two models. We're going to do that by visualizing individual sites that are scored differently. So we're going to create plots that look like this. On the x-axis, I'm going to put the prediction that the mouse model gives for a site, and on the y-axis, I'm going to put the prediction that the human model gives for a site. We're going to plot all of the sites in the human test set this way. So sites that are going to fall along the diagonal will be wherever the models agree, and sites that fall in these corners will be the interesting stuff that will be where the models disagree. And so if we first look at the bound sites in our human test set, what you can see right away is that all of the true positives are up in the top right corner. Those are sites where the models both agree that the site is bound, and indeed that is the truth. In the bottom right corner, for some TFs, we can see some enrichment. There are some false negatives common to both models, but most interestingly, we want to look at the top left corner, which is where false negatives unique to the mouse model would be. And while we see that there are some sites in those corners, generally it seems like false negatives unique to the mouse model might not be a huge driver of the AUPRC gap that we observed. On the other hand, if we look at the unbound sites from our human test set and look at the bottom right corner where we would find false positives unique to the mouse model, we see that there is indeed a large enrichment of sites, particularly for HNF4A and RXRA. And so it seems like these false positives might be a stronger driver of that AUPRC gap. Now you might be asking, is there anything special about those sites? Do they all have something in common, some genomic annotation that overlaps many of them? And the answer is yes. So what we did was we took these plots of unbound sites and we filtered them to only show the unbound ALUs, which are a type of repeat element in the human genome. And what we saw is that overwhelmingly the sites in that bottom right corner, the false positives unique to the mouse model, they are ALUs. And so that is our culprit. So we can say that the mouse model is systematically over-predicting unbound ALUs when it tries to transfer to testing on the human genome. And it's not surprising that this is happening really because ALU elements are unique to primates. And so the mouse model just never saw them during training, which sort of begs the question, is this an issue with generalization across species, or is this an issue with the human genome being somewhat different between the not ALU repeats and the ALU repeats? right? Because the mouse model didn't have as much trouble generalizing to sites that weren't ALUs in the human genome. And so what we did was we tested out trying a new human model that was trained on the same data set as our original human model, but we removed all the ALU repeats from the training data. And so on the right, that's what I'm showing. The x-axis is now the prediction from this human model that hasn't seen any ALUs. And what you see is, although there's great concordance between the two human models on the bound sites, on the unbound sites, we are again seeing this off-diagonal enrichment in the bottom right corner of false positives that are unique to the model that hasn't seen ALUs. And so that does seem to be the problem. So to investigate what are the actual sequence determinants that are causing this, like more seriously what is going on here, we turn to importance scoring, which is a strategy we can use to ask a given model which bases in some input sequence were important for the prediction that you gave that input sequence. And so we're going to do this for both models, and then we're going to see to what extent do the models differ in their importance scoring when they also differ in their predictions. But first, we're going to just look at some true positive sites as a control, sites that were bound and that both of the models agreed were indeed bound. And so the importance scores look like this for a couple examples, one for each TF. And what you can see is that we are very easily able to see motif agreement between the importance scores for both models. Generally, the bases are weighted pretty concordantly across the two models, and we are very easily able to recover the canonical PWM representation of the motifs for all of these TFs in these sites. And so it does look like on sites where the models agree in prediction, they do also agree in importance scoring. And importantly, it doesn't look like the models from two different species are learning two different motifs. Generally, they seem to be looking for the same thing in these sites. On the other hand, if we look at the interesting sites, those mouse model false positives, we see something totally different. And so I've zoomed out so that you can see the noise that's present in these scores. And there are two different interesting things going on here. So first, it seems like the mouse model is learning these pseudo-motifs, these small chunks of highlighted sequence that may or may not be matches to parts of the PWM representation of this TF's sequence affinity, but also that there are select bases in these sequence that the human model particularly downweights as evidence that it should not predict this site as bound. And the mouse model, meanwhile, seems to be completely unaware that these bases should downweight its prediction. And so it does seem like there are specific sequence features that the human model knows not to predict this site as a bound site because of. Another way of looking at this or answering the question, what's going on at the sequence level that is causing this false positive issue for the mouse model, is to look at subfamily enrichment of specific ALU subfamilies. And so I'm going to define enrichment score, which is just the fraction of mouse model false positives that overlap some ALU subfamily divided by the fraction of both model false positives that overlap that subfamily. And to recap what those site terminologies are, so the both model false positives are unbound sites that both models mispredict, and the mouse model false positives are unbound sites that only the mouse model gets wrong. The human model gets it correct. And so if we calculate this enrichment score across all the ALU subfamilies and across the 4 TFs, we do see really interesting patterns emerge. So it seems like things are split between the ALU Js, the ALU Ss, and the ALU Ys. The ALU Js seem to not be very enriched, which indicates that they might not play a huge role in this mouse model false positive story, whereas the ALU S's, with the exception of CTCF, seem to, across the board, be pretty fairly enriched in the mouse model false positive. So there's something consistent between the sequences of all the ALU S repeats that is causing this problem. Most interestingly, the ALU Ys have this heterogeneity going on, where across TFs and across subfamilies for the same TF, there's huge differences in the enrichment scores within these mouse model false positives, indicating that some of these subfamilies have the sequences that cause the false positive issue and some of them don't. So what's causing it is probably something very sequence specific. Now, if we switch from descriptive to prescriptive and try to think about how we might actually fix our mouse to human models so that they do better, we can frame our mispredictions on these species unique ALU repeats as a domain shift, where our domains are the mouse and the human genome respectively. If we do that, we can ask the question, can domain adaptation help our mouse to human models? A quick recap into what domain adaptation is if you're not familiar. So if we visualize our human and mouse genomes as being blue and red respectively and embed them in the same space along the same axes, and we color our bound sites as squares and our unbound sites as circles, if we were to train a classifier on our mouse mouse genome on the left, it would draw the green line that you see there. But if we were to then just apply that classifier naively to our red domain, what you would see is there would be mispredictions happening because of that domain shift, because the squares and the circles are not aligned on these two subspaces. So the goal with domain adaptation is to align the distributions of these two domains so that when a classifier gets trained on only one of the domains, it just naturally applies well to the other domain. And in particular, we're going to be pulling from the work of Gannon and Lempinski, who say that for effective domain transfer to be achieved, predictions must be made based on features that cannot discriminate between the training and test domains. What that means for us is we're going to want to have a model that makes predictions based on sequence features that can't discriminate between our two species. So to return to our model architecture and training strategy, this is what we had before. So we've got one training set, and it's got sequences that are either labeled as bound or unbound. Those propagate through the model, and then at the end, a prediction comes out whether or not a TF is bound to that site, and backpropagation is what encourages all of the weights in the model to learn discriminative features that can help distinguish between the bound and the unbound sites. So what we're going to do is we're going to add a second model output that is going to predict what species the input sequence is from. And I know that without modifying our training set, this doesn't make much sense, but bear with me. So as this model is designed, this is probably going to do exactly the opposite of what we want it to do. Instead of learning features that cannot discriminate between species, as Gannon and Lempinski advised, this is going to train a model that will simultaneously learn binding and also be as discriminative between species as possible. And so all we have to do in order to flip that process is to add a gradient reversal layer in between that secondary task head and the convolutional filters below it. And so what's going to happen is as this model trains, the backpropagation will encourage discriminative features that distinguish between bound and unbound sites, but it will discourage discriminative features that distinguish between mouse and human sequences. And the only reason is because when the loss backpropagates through that second task head, it's just going to be multiplied by -1, and that means that the weights in the convolutional filter will be updated in the opposite direction that would be helpful for species discrimination. So I said we were going to have to adjust our training sets here. So the updated training scheme is that we're going to have a second training set. It's going to be 50% random sequences from the mouse genome, 50% random sequences from the human genome, and then we're going to split our training procedure into 2 stages for every batch. So in the first stage, examples from the first training set are going to propagate through the model. They're only going to go through the TF binding output, and so the loss will only backpropagate from that head back through the convolutional filters. In the second stage, we're going to take examples from our second training set, and we're going to propagate them only through the species discriminator secondary model output. And so very importantly, the TF binding classifier task head will never see human examples, and it will never see binding labels from those examples. So this is still unsupervised domain adaptation. We're just using background distribution sequences to inform our model. And so the goal with this is that as this trains, the model specifically unlearns any sequence features in the convolutional filters that could discriminate between human and mouse sequences, including, notably, ALUs. So how well does this work? Well, if we repeat our procedure as before, training our mouse models with domain adaptation, as shown in green on these plots, what you can see is for 3 out of our 4 TFs, we do indeed see that there's a performance improvement according to AUPRC. And you'll note that CTCF was a TF where the ALUs weren't as enriched in the false positives as they were for the other TFs, and so that's probably what's going on there. Correspondingly, we don't see much of a decrease in performance on the mouse test data for those same models, indicating that we haven't totally ruined or sacrificed a whole bunch of our performance on our original domain when we've applied this procedure. But what we really cared about was the ALUs, right? And the AUPRC prioritizes performance on the bound sites, and so this might not be the best reflection of whether or not we've actually fixed the whole problem. We do know that there's still a gap here between the human trained models and the domain adaptive models, but the real question is, did we fix the ALU problem? And so if we return to the plots from before, for two of our TFs here, we were plotting our unbound sites, and so we saw an enrichment in the bottom right corner, and when we looked at just ALUs, we saw, again, bottom right corner, pretty much all ALUs, and enrichment of sites there. If we apply domain adaptation, these plots instead look like this. And so what you see is there's a large decrease in the number of sites that are hanging out in the bottom right corner there. And in particular, on the ALU plots, you can see that there's been a massive migration of those sites that were hanging out along the x-axis. They've all shifted left because the mouse domain adaptive model is doing a much better job of not predicting them as bound when they aren't. We could look at the same thing for the other two TFs. Again, this is the before and this is the after with domain adaptation. And again, it really looks like the ALU problem has pretty much been solved in this case because that bottom right corner is devoid of ALUs. And so in summary, what we did was we tried to predict transcription factor binding across species and found that the models suffered from a major performance gap. We found that that gap was caused in part by systematic misprediction on ALU repeats that are unique to the test species or the human genome. We then performed this training integrated strategy of unsupervised domain adaptation, which discouraged the neural network from learning those species-specific sequence features, and we found that it did improve cross-species TF binding prediction by correcting those mispredictions. So to conclude, I'd like to thank both of my labs, both of my advisors, everyone who helped on this project, and our funding. And with that, I'd love to take any questions you have. Thank you.